talk about the biological concepts, introduction, model, methodology, results, and summary. First of all, fitness means an organism's ability to survive and reproduce in environment and passes its genetic material to the next generations. And obviously, an organism with a higher fitness passes more copies of its genes. Mutations can lead changes in the phenotypic characteristics of individuals, and mutations can have a potential either increase, decrease, or do nothing to fitness of the mutated individuals. When a mutation changes from wild-type individuals to the mutated, the fitness effect of the mutation can be denoted as the S, which is the fitness of the mutated over fitness of the wild-type -1. Next concept is distribution of fitness effect, means the probability distribution of the fitness effect of all available mutations for a specific ancestral organism. And so this figure shows the DFV schematic, and the x-axis is the selective effect of the mutations. When the fitness of the mutated individual is bigger than fitness of the wild type, S is positive, mutations are beneficial. When this fraction is equal to 1, S is 0, and mutations are neutral. And when W mu is less than WW, S is negative, and mutations are deleterious. So distributions is the fraction of these mutations, DFE. So mutation accumulation experiment is a type of biological, is a type of evolutionary, experimental in evolutionary biology. So an ancestral cell is divided into the n replicated lines, and each line a mutation can happen. They can become fixed or lost during the generations. And after n generations, mutations are allowed to accumulate randomly without any selection pressure. But now the question is why the mutation MA experiment is important. In order to get more accurate measurement of the DFV, the effect of the selection must be minimized. MA experiment tried to eliminate the selection effect by doing a serial population bottlenecks. So this is the original population. At the bottlenecking event, some populations are chosen at random. After a passage of the time, we have a surviving population. And in the next generations, obviously, we have more red, less blue, and a few number of the green. This is the general population bottlenecks. But as you know, we have a different sizes for the bottlenecks. In our studies, the size of the bottlenecks is 1. So we choose 1 individual at random to reproduce, and there is no selection in the bottleneck stage, but the selection and competitions between wild type and mutated individuals happen during the growth. So in large organisms like birds and flies, it's not difficult to achieve a bottleneck of 1 or 2 individuals. However, in microscopic species like bacteria, it's not possible to do the bottleneck. So we need visible colonies, which the multiple generations are required between bottlenecks. For bacteria, between 15 to 13 generations provide enough time for beneficial mutations spread throughout the bacterial colony. And even if we only select 1 individual at random, biases are introduced due to the selection during the growth between the bottlenecks. In a study, the experiment, which is done by Deepa, our collaborator, they take a sample of individuals and they add a lot of water to wash them, and cells be far from each other. And on the new plate, each colony started with a single individual, and so single individuals started growing to make their own colonies. So after having this colony, they choose a colony at random to transfer to the new plate. When they're comparing the beneficial and deleterious mutations that happen in the chosen colony, beneficial mutations represent more higher frequency, and this causes the underestimation of the deleterious mutations. So in the previous work, which is done by Deepa and Lindsay, my supervisor, the effect of the selection in a MA evaluated by deterministic model. They started with a single individual. It grows exponentially, and mutations happen per individual per unit time. And they grow exponentially, and with assumption that the mutations are not lost from the population. However, in reality, the number of offsprings and individuals contribute is random, not deterministic. A mutation can be lost and no longer exist at the time the colony is sampled. So in order to take this into account, two offspring distributions modeled by using the probability generating functions. And when there are multiple generations, we use the branching process. Let's talk about my model. So let X and Y be a dummy variable for wild type and mutated individuals. And Pij is the probability that the wild type contributes I wild type and J mutated individuals. So this formula one is the PGF when we are starting with the single wild type individuals. Similarly, we have a PGF when we started with a single mutated individual. So the PGF for the number of individuals in the first generation is just Fxy. And because the independence of the individuals in branching process for the more than one generation, in order to get the PGF, we use the composition function. For example, after tau generations, PGF for the individuals of the tau generations is the composition F by G tau time. So if I expanded this expression into the polynomial form, we will get this equation. And these coefficients are a state probability. For example, P1, 0, and tau is the probability of having one individual, one wild type individuals and 0 mutated after tau generation. So now we want to apply the branching process in our microbial lineages in which the offsprings follow fission distribution. So we have a wild type cells that is divided into the two equal dotted cells. Each cell has a probability of dying before reproducing, and PGF for the surviving offsprings is like both of them die, only one of them survive, and both of them survive. And so each daughter cell has a probability of carrying mutations. So PGF of the mutation process is with the probability 1 minus mu individuals are wild type and with probability mu are mutate. So PGF by composition F1 and M, we will get the PGF for all surviving of individuals, both wild type and mutated. So when both of them are survive, so we have three cases, both of them are wild type, one of them are mutated, the another is wild type, or both of them are mutated. So let's detail that is the death probability of the mutated individual. So selective effect or selective advantage of this mutation is equal 1 minus d tilde over 1 minus p minus 1. And if d tilde is equal to 0, we can find the maximum possible selective advantage. And so when we are starting with the mutated individuals, the PGF for the offspring is like when both of them die, only one of them survived, another one died, and both of them survived. So now we want to compute the selection bias. So in order to get the relative bias, we need the probability that individuals carrying a mutation with effect S is selected. So it's like equation P of S. So PMN is, we have a M wild type individuals and mutated. So the probability that the mutated is chosen is N over N plus M times PMN as an example. So the relative bias is equal to probability that a mutated individual with effect S is chosen over the probability that the same individual with effect 0 is chosen. So we are looking for the formula to find the P of S. So we use the analytical method to get the relative bias. We take the first derivative of the PGF after tau generations with respect to the y, and then substitute y equal to x, and then we integrate from equation 8 with respect to the x from 0 to 1, and so we get the P of S. As a reminder, P of S is this equation. So we are able to get to find our relative bias. So let's talk about the analytical result. This plot shows the relative bias plotted versus selective effect S. So for S bigger than 0, the mutations have a higher relative bias beneficial mutations for different number of generations from 5 to 27. By increasing the number of generations, the relative bias for beneficial part is increasing, and these represent the same data on the semi-scale plot, and so these are for deleterious part. By increasing the number of generations, the deleterious is decreasing. So this is for pure burst model, the deterministic model. So we have a stronger the selection effect or relative bias when we apply the fission distributions on our model. So you can see when the offsprings follow the fission distributions, we even have a higher relative bias for beneficial part. This figure compared the selection bias for different death probability and comparing the pure burst model with fission distributions. So again, for when S is positive for beneficial part, the fission distributions and shows the higher relative bias compared to the pure burst model. And these are again the same data on the semi-log plot. And when we increase this probability, we can see even more relative bias. And for both panel A and B, for the deleterious part, when S is negative, the relative bias for the deleterious part is for fission distributions is always lower than the pure burst model. Okay, so then we simulated our experimental data in MA experiment. So if there is no bias, we expect to have a DFE like this as a true DFE, but when we simulated our data, we observed the DFE like this, which is shifted toward the beneficial mutations due to the selections during the growth between the bottlenecks. So after applying our corrections to the model, the corrected DFE shows an excellent agreement with the true DFE. So the corrected DFE in the blue histogram, it's very shows a very good agreement with the true DFE. These are for fission distributions 27 generations. We did all of these analysis for Poisson distributions, but because of the limited time, I just talk about the fission distribution. So as a summary, we developed a fluid stochastics model that incorporates the realistic distributions of bacterial offsprings, and we demonstrate in our model that the biases caused by selections on the observed DFE is even stronger than the biases predicted in a pure burst model. And so the correcting for the selection bias in MA is even more important than previously argued. Just please, thank you for listening.
Thank you, Mutan. We have a little time for questions. Any questions? I can have one short question. So do you have intuitive explanation for why fission is making the selection bias stronger? To understand why?
Because in the bacterial cell is divided into the two cells, we use the fission distribution. Fission distribution is like the binomial distributions when n is equal to 2, because it's more close to the realistic. We choose the fission distribution.
Okay, thank you. Maybe in the meantime, you can stop sharing your screen and we can have Katie McManus sharing your screen. There are other questions, maybe in the Q&A. No. All right, so we start with Katie McManus talking about phenotypic divergence associated with urbanization in the common butterfly. Thank you.
Thank you. So one of the most profound drivers of anthropogenic change is urbanization, and urbanization is one of the fastest growing land use types. It's predicted that global urban land cover will at least double by the end of the century, increasingly housing more of the world's biodiversity. And when we talk about urbanization, it's important to define what we mean by urban. So these are areas that have more than 50% of the landscape covered by impervious surfaces, and they have co-occurring high levels of human activity, such as high human population density. And urbanization produces unique landscapes that have a unique combination of selective pressures. These include increased air pollution, altered levels of nutritional resources, increased immune and metabolic stressors, higher temperatures due to the urban heat island effect, and barriers to dispersal. And this begs the question of how urban populations will adapt to survive. So to begin to understand how urban populations are adapting, I'm examining the patterns and potential drivers of phenotypic divergence in traits related to flight and pigmentation in the cabbage white butterfly. And specifically, I ask if flight and pigmentation traits differ between urban and non-urban populations, and if these traits vary along a gradient of urbanization measured by temperature and impervious surface. So I'm using Pierre's Repet, the cabbage white butterfly, as my study system. This butterfly was introduced from Europe to the United States around 1855 and has been here since the Industrial Revolution and the growth of cities. The cabbage white has a ubiquitous cosmopolitan distribution across the United States, likely due to its broad environmental tolerance. And the cabbage white uses plants in the Brassicaceae family as their larval host plant, and the adults will use various nectar plants. And the species is sexually dimorphic, where the males shown on the left here are slightly larger in size and the females are more pigmented than the males. The females also display this additional spot on the forewing. So I sampled cabbage whites across 3 regions in the eastern United States. Within each region, I sampled at 3 to 4 urban sites and 3 non-urban sites, and I collected both males and females to see if we're seeing different responses in the sexes. I collected an average of 26 individuals per site for a total of over 500 individuals caught. And for all individuals, I collected trait data on traits related to both flight ability and heat load. So I obtained a dry mass for the body and for the thorax, and I measured the forewing area. And if fragmented urban habitats are selecting for increased dispersal capacity, we may see increases in these traits. So increased body mass and thorax mass and an increase in forewing size. However, in some species of butterflies, fragmentation has been found to select against dispersal. So it's also possible we may see a decrease in body mass and thorax mass and a decrease in forewing area. Additionally, it's also possible that the higher urban temperatures are selecting for decreased body size and decreased wing size, as this would allow for more heat dissipation and prevent overheating in urban areas. So for each trait, I modeled the effect of urbanization on the trait in 3 different ways. So each figure will show the metric of urbanization on the x-axis with the trait on the y-axis, and I'll show the data separately for females in red and males in dark blue. So first, I used urbanization as a categorical variable and classified each site as either urban or non-urban. And I found that the urban butterflies have less total mass. So we see that the urban males are smaller than non-urban males, and the same comparing urban and non-urban females. But because within the categories of urbanization, as urban or non-urban, there could be a lot of differences. I also modeled urbanization using continuous predictors that could be driving the responses, namely impervious surface and summer temperature. And for both continuous predictors, we see the same trend that with increasing urbanization, there's a decrease in total mass for both sexes. We also found that the urban butterflies have smaller thoraxes, and that this holds true again for the continuous measurements of urbanization and across both sexes. However, when we look at forwing area, we only see an effect of urbanization in males. So we found that the urban males have smaller forewings than non-urban males, while the urban females don't differ from the non-urban females in forewing area. And we see the same trend with our continuous predictors. So with a negative slope for males indicating a decrease in forewing area with increasing urbanization. And from my measure of body mass and forewing area, I calculated wing loading. So this is an important trait related to the aerodynamics of the individual. And it's especially important for maneuverability and will determine how fast a butterfly can take off. So a decrease in wing loading would correspond with an increase in flight performance. So if dispersal is selected for, then we would predict that the urban individuals will have a decrease in wing loading. If dispersal is selected against, then the urban individuals may have an increase in wing loading. And when we look at the data with these categorical predictors, we don't see any difference in wing loading that's driven by urbanization. We purely see sex differences. However, the continuous models are able to capture an effect of urbanization. And we see that for both sexes with increasing urbanization, whether we're using impervious surface or temperature, there's a decrease in wing loading. Lastly, I calculated wing brightness at wing spots that are related to thermoregulation for this species. So adults of this species can control their body temperature through the combination of the pigmentation on their wings used with different basking behaviors. So the melanized wing scales that are close to the body in this area called the discal cell will absorb solar energy, and it can conduct heat directly to the body when they're in this dorsal basking position with their wings flat out. Therefore, in warmer climates, such as urban areas, this area may be less pigmented so that it can reduce solar absorption and prevent overheating. Similarly, the ventral hindwing can absorb solar energy when it's in this lateral basking position where it has the wings are closed and this position is exposed to the sun. And this also can directly conduct heat to the butterfly's body. So this area may also be less pigmented to prevent overheating. The wing spots that are further from the body function in a different way. So these areas are involved in reflectance basking. And this is where the butterfly will have their wings at this 5 to 90 degree angle, and solar energy will be reflected off of the wings onto the body. So I'm interested in these 3 melanized spots that will be used in reflectance basking, and the amount of pigmentation in these areas will impact how much reflective surface remains. Therefore, these spots may have increased pigmentation in warmer areas, which will allow them to decrease that solar reflectance. And interestingly, when we look at pigmentation, we don't see an effect of urbanization on pigmentation with insects. So urban males have the same pigmentation as non-urban males. But what we do see is an increase in sexual dimorphism in the urban populations. So for the wingtip, we see a difference between urban males and urban females that we're not seeing in the non-urban population. And when we look at this with our continuous predictors, we can kind of see what's going on. So we see that there's a slight increase in brightness for the males with increasing urbanization and a slight decrease for females, which is driving the signal of sexual dimorphism that we're picking up on. And we see the same trend with ventral hindwing brightness, where the males are increasing in brightness, which is driving sexual divergence and dimorphism in urban areas, and we don't see it as strongly in non-urban areas. And the remaining wing pattern areas didn't show any effects of urbanization. We see trends that are in line with what we already know based on the sexual dimorphism of the species. So the upper and lower wing spots and the discal cell are all darker in females, which is in line with what we know in natural populations. And I don't show the results of the continuous models here, but they also don't find an effect of urbanization. So same results as here. So overall, when we're predicting outcomes to urbanization, we see similar trends, whether we're using the categorical or the continuous predictors. However, for all the traits in this study, the models that had that simple urban, non-urban comparison were best when we did model selection with AIC. So from this, it seems like it's not enough to capture just one axis of urbanization, such as only impervious surface or only temperature. So the categorical classification can capture more of those urban stressors. I'll also note my model included a random interaction between urbanization and region, and we did not find a significant interaction. So this indicates that the responses are the same across the different metropolitan regions that I sampled in. So from these results, we can't tell whether these are the result of plasticity, an evolved response, or possibly breeding depression. But regardless of the mechanism, there are potentially important ecological consequences from these changes in urban areas. So first, the decrease in body mass in urban butterflies is important, as body size is correlated with many aspects of life history, especially reproduction in females. So in females, the body mass is positively correlated with egg production. So it's possible that urban females may produce fewer eggs than the non-urban females. Additionally, body size is also one aspect of morphology that has been found to be correlated with flight ability in this species, specifically flight speeds. So it's possible that the urban butterflies may be slower flyers. For both sexes, we also found smaller thoraxes in urban areas, and this could also lead to decreased flight ability, specifically for their acceleration and their endurance, as this is the area that contains the thoracic muscles for flight. For urban male butterflies, we found that they had smaller forewings than non-urban males, and forewing size has been found to be positively correlated with flight performance in butterflies, and this correlation is especially strong in males, and this relationship may be especially important in males as well because the male butterflies do spend most of their active time actively flying, searching for mates, so changes in flight ability could have a large impact on reproductive success. And on the other hand, though, wing size is also related to thermal heating rate in butterflies. So urban males may be better at dissipating that excess heat and reducing their heat load in warmer urban areas because of their smaller forewings. And so while we're seeing potential negative implications in those first three traits in regards to flight ability, there's also a co-occurring decrease in wing loading that could result in improved flight performance, specifically for maneuverability. Lastly, we also found an increase in sexual dimorphism of pigmentation in urban butterflies, and pigmentation is thought to be important for mate choice and species recognition in the species, so this may also have reproductive implications. With that, I would like to just thank the Spigler Lab in the Department of Biology at Temple, as well as the different bureaus and landowners who let me sample on their sites, and this was funded by the American Philosophical Society and the Entomological Society of America. And I will take any questions if I have time.
Thank you, Katie. Any questions? Maybe I'll start with one. So you have two different continuous predictors of urbanization. And I was wondering whether it was really possible to separate the effects or whether they were co-varying as well, because I guess temperature is also increasing when you change the type of surfaces you have. So how can you tease apart really the effect of those, and does it shed a different light on your result if you do?
Yeah, so they are both highly correlated, and we do see similar trends for both. So it is possible that we're not seeing effects of impervious surface, we're seeing effects of temperature. I think from just the observational data, it is a little more difficult. It would take more like experimental work to see, is it temperature that's driving these changes, or is it impervious surface, if we're thinking one of those is the driver.
And so let's see. Are there any questions? There's 1 minute left. Maybe the next speaker, who is Jesualdo Arturo Fuentes Gonzalez, can share his screen, and we have a last question for Katie. Actually, I had another question. Did you try to have like more direct test of dispersal, you know, not morphology, but behavioral test of dispersal propensity or behavior in your butterfly to test the idea that it's really linked to, because it's sometimes hard to connect those morphological traits to actual dispersal?
Yeah, right now I only have those morphological traits because I just have the captured and like pinned and spread individuals. But it would definitely be cool to have live individuals and be able to see differences in flight ability.
Okay, thank you very much. We can now start with Jesualdo.
Okay, thank you very much. Thanks everyone for being here. Very excited to give this talk to you. I want to start by acknowledging my co-authors, which were really critical to make this work possible. So the morphospace is a really useful tool to study macroevolutionary patterns. And in geometric morphometry, they're particularly helpful because they have visualization capabilities that aid in interpretation. To see it more clearly, let's explore this morphospace in canine. This is particularly a canine jaw. So we place some landmarks that are shown here in the image. And after going through the standard geometric morphometrics procedures, we obtain a morphospace where each point is representative of a species. So the cool thing is that we can identify the distribution of those points, reveal some patterns. So for example, in this case, we find that some specific diets, for example, carnivory, which is shown in blue, and non-carnivory, which is shown in green, we are going to refer to these like basically as omnivory for simplicity. They occupy different regions of the morphospace. So the advantage here is that we can identify, okay, is diet related to the morphological attributes that we see in this morphospace? So another cool thing about the morphospace in geometric morphometry is that each point in it is associated with a specific shape. So here in orange, I'm highlighting this orange point in the center of the morphospace is the consensus, which is a reference shape that captures the variability in morphology in all the sample. So you can see in this grid, these points are the same ones that we see in the image. And basically, they represent variability in the position of those points in the structure. You can see in these grids that basically there are perfect squares indicating that it is non-differentiated relative to other places of the morphospace. Any other point in the morphospace relative to that consensus is going to work in some way. So, for example, if we explore the extreme that is associated with carnivory, and we compare it with the extreme that is associated with omnivory, we can see some differences in those shapes. We can see here, for example, that the main difference is in the modal region. So carnivorous shapes have a reduced modal region compared with the omnivorous ones, which makes sense functionally because a reduced modal region indicates a shorter jaw, which indicates like stronger bites because there is higher mechanical advantage. And the increased molar region, we can interpret that in terms of more grinding area for processing food. So it is very tempting to interpret these differences in terms of adaptation. The issue is that if we think about it, the evolutionary process underlying adaptation is natural selection. And natural selection works by reducing variation. So there is not a priori reason to think that morphologies at the extreme of the morphospace should be favored relative to morphologies in the center of the morphospace. What this reveals is that there's sometimes a gap between the patterns that we see in the morphospace, how to want to analyze them, and the evolutionary processes that generated those patterns in the morphospace. What I want to show you today is that there are ways to fill this gap, which basically reduces to having a gap between macroevolution and microevolution in the study of morphology, and that the adaptive landscape is a helpful tool that can help us to close that gap. So since the very beginning, the adaptive landscape, as proposed by Wright, has a strong emphasis on selection. The issue is that it is not really helpful to study the morphospace because it's very focused on genotypes and populations. But later on, Simpson expanded the same idea for phenotypes and higher taxonomic levels, which applies better for the morphospace. Simpson inspired two lines of work that I would like to mention today, several lines of work, but I'm going to focus on two. One are the fitness surfaces that were proposed by Landy. So these use population parameters and a mathematical model that basically balances the effect of selection and drift. The advantage of this is that we can model specific evolutionary processes in this framework. In principle, it is designed to model stabilizing selection. But if we modify the topology in that equation and how it changes through time, we can model other processes too, such as neutral evolution, fluctuating selection, and adaptive evolution. The other framework that I want to mention are phylogenetic comparative methods, especially those grounded in evolutionary quantitative genetics. And I want to focus on the adaptation inertia method proposed by Hansen, which uses historical context and an Ornstein-Uhlenbeck model to explore different evolutionary hypotheses. So for example, here we are seeing historical context informative of diet at the left, and in the right, while there is informative of cladogenesis. And we can test, for example, which of these two is more likely to have affected, let's say, a morphology in the case of the morphospace, in terms of adaptive evolution. And we can contrast those hypotheses with simpler ones. For example, an Ornstein-Uhlenbeck in which there is only one peak, which is, for example, informative of constrained evolution, or one in which there is no regime at all, which is basically reducing evolution to a Brownian motion process, which is valuable, for example, to model neutral evolution. So the thing here is that the fitness surface framework gives us a solid ground to test evolutionary processes, and the comparative analysis gives a framework to test interspecific data. So the idea is that if we can find a way to merge those two approaches for the study of the morphospace, we have an opportunity to fill the gap between micro and microevolution in the study of the morphospace. And this is what we wanted to do here. We did that through simulations. Basically, the idea is simulate data in the morphospace based on the fitness surfaces of Landy, and then test if those surfaces with comparative methods could be recovered with the specific models. So the way we did this, we used the literature to gather different population parameters in canites, so that the simulated morphology that we were generating were relevant for the evolutionary processes that we were interested in, that are the ones that I showed you earlier. We did that in the tangent space of that jaw morphology that I showed you earlier. So basically, the simulations that we are doing in the morphospace are relative to these jaw morphologies. So for the peaks, in models with a single peak in which we were using selection, we used the consensus as the reference point. But for those having more than one peak, the extra peak was modeled based on the fossil record, in particular, the dire wolf. And then what we did is let the phylogeny evolve on those landscapes. That gave us interspecific data that we were able to test with the phylogenetic comparative methods. We did that like many iterations with different parameterizations. The idea was to see if there was consistency in the models that we could recover associated with those landscapes that I showed you earlier. So what we see using information criteria and accounting for the complexity of the models is that indeed there are patterns. Basically, there are specific models that are favored depending on the evolutionary processes. When we have smooth surfaces, basically surfaces with no noticeable peaks, the preferred model is Brownian motion, which is consistent with what we see in the univariate literature. Now, when there are noticeable peaks, Ornstein-Uhlenbeck is a better explanatory model. And when, for example, stabilizing selection does a better job in explaining, but the Ornstein-Uhlenbeck with a single peak does a better job explaining stabilizing selection. And the two peaks do a better job at explaining adaptive evolution, particularly the diet regime. So the cladogenetic regime was not really favored because it was basically a control regime. It was not supposed to be associated with any process. So now that we know that these models actually can capture evolutionary processes that happen in the landscape, what happens with the real data, with the real canine data set that I showed you earlier? So what we see is that the favored model is the adaptive regime. This gives us an indication that indeed feeding preferences have had an effect on the evolution of jaw morphology in canites. And the cool thing about this is that we can actually map where those peaks are in the morphospace based on the results of the model. So here we see all these closed dots are the canine species differentiated by color in terms of diet. The omnivorous again are in green, the carnivorous are in blue. These triangles are the adaptive zones, basically those adaptive peaks in the morphospace associated with the diet. And the cool thing is that I'll show you, we can see the shapes associated with those peaks. And we can contrast them with the extremes in the morphospace that I showed you. The interesting thing here is that if you look closely, even when the shape, the optimum for carnivore looks kind of similar to the optimum for to the extreme in the morphospace, the one for omnivory doesn't look so close to the extreme in the morphospace that we showed earlier. And that makes sense because if you see, this peak is actually very close to the consensus, not so close to the extreme. That means that you compare the two shapes too, and you can see that this omnivorous shape is not very differentiated. And this has evolutionary relevance because basically it tells us that omnivory, which is the basal diet, is probably more affected by stabilizing selection and maintained on a generalistic form than carnivory, which is the derived diet. So basically that one has worked more due to functional adaptation. So notice that even when the extremes in the morphospace give us like a good visualization way of exploring patterns, the model that I'm presenting you here not only capture that visualization advantages, but are more grounded in evolutionary processes. Actually, I will argue that even in terms of the visualization, this approach performs better. And the reason is that look that if we focus only on the extremes, it is very likely that we're only captured by these axes of the morphospace and ignore the other axes. And actually, the optima that we observe are very in both axes. So we are gaining a little bit of extra information when we look at this optimum, and we can see that comparing this with the canine jaw. Look that the optimum gives us extra information on the diastema, which are these points here, and the thickness of the jaw. The diastema, the reduction of the diastema implies even further shortening of the jaw, which is even indicates like higher mechanical advantage and more biting stress. And the thickness of the jaw is informative of a more robust jaw. Basically, it is advantageous for capturing prey that are struggling, and so it is more resistant to mechanical stress. So look that these attributes are very captured by the optimum. Basically, it is information that is distributed in both the X and the Y axes. So if we ignore that Y axis, we cannot see it, and it is very easy to ignore that axis when we only focus on one specific extreme of the model. I want to emphasize that my point is not in criticizing the visualization tools of geometric morphometrics. They are very useful and I use them all the time. What I wanted to emphasize is that if we want to evaluate the morphospace in a more grounded evolutionary setting, using like models that are more consistent with all the evolutionary processes are more helpful to fill the gap between pattern and process, and in this way, fill the gap between micro and macro evolution. And with that, I want to thank the several institutions and people that have supported this work, and you, of course, for listening. And if I have time, I'll take questions.
Thank you, Resueldo. Just on time, so we have a lot of time for questions.
Actually, I have a question. Did you see? I see a lot of Zoom stuff in my screen that was getting in the way. Do you see that stuff too, or did you see that in the talk, or it was only me seeing these things?
What did you see in your talk?
Like the different little screens like associated with the Zoom room. But I don't know. Did you see only the slides? Because I was seeing like
No, we saw just a slide. Don't worry.
Okay, I'm glad. I was just like moving stuff all the time.
No.
I was getting in the way.
So I guess maybe I have a question. So those comparative methods, those trying to fit those Ornstein-Uhlenbeck models and stuff, they were also criticized because there was a lot of kind of false positive or stuff like that. So is your approach immune to that, or is it?
Sorry,
like,
yeah, immune to that? Could you repeat that?
Like sometimes you can select a model for reasons that are not really related to micro evolution, or you might find signal for attraction to a different optimum and stuff like that, but they are a bit spurious. I know there have been some debates about that, but
Oh, got you. Yeah, no, actually, that's good question, and I hope a part of the idea of fitting other models, the idea was to say something about that. Notice that if it was true that the complexity of the model tends to be favored regardless of what was the process. Notice that we will have consistently obtained more of those complex models favored in the study of the morphospace. But check, for example, that when we were modeled simple hypotheses like stabilizing selection, those more complex models, those peaks were not selected. And for example, know that we modeled two specific regimes that were complex, one associated with diet, which was the one that we actually modeled, and was that was not associated with diet, but that we fitted just to see the effect of that. And the one that was selected was the one that actually simulated in the morphospace. And the idea of adding that control was testing that possibility.
yeah.
So,
Thank you.
Thanks.
Maybe Parnian Bahami, you can share your screen. No?
Okay. Hi everyone, my name is Parnian Bahami from Dr. Singhal Phage Lab, and today I'm going to talk about genetic evolution of 5-6 undergoing different heat shock treatments. So the climate change leads to increasing of the temperature, and global warming has several impacts on different aspects. Some of the impacts that I can name are our organism, ecosystem, health, social, economic, and agriculture. But what if we want to know what would happen to the organism in terms of the temperature is rising rapidly? So maybe some of the organisms, they can tolerate higher temperature. And as another option, maybe some organisms, they can move to a more appropriate environment. And if in case the organisms, they can tolerate higher temperature or they can move to another environment for aiming their survival, we expect them to evolve. And in the procedure of evolution, they need to fix some mutations. So we are answering this question with the help of bacteriophages, which are the viruses that only infect and replicate in the bacterial cells. The reason that we are using bacteriophages is because they have large population size and they have very short replication cycle, and also they have high mutation rate. So our specific study system is 5-6 bacteriophage, which is from Cytoviridae family. And it is the first known double-stranded RNA virus, and it has a membrane around the protein coat, which is originated from the host that this virus infects. This virus replicates in the room temperature, which is 25 degrees Celsius, and the host of this virus is gram-negative Bacterium pseudomonas syringae. Our research question would be if 5-6 bacteriophage can tolerate the heat shock treatment, and if they survive or not. And in case if they survive, we want to detect those mutations that cooperate in their surviving, that we are calling them thermostabilized mutations. So the research question would be, how does 5-6 bacteriophage adapt to temperatures that increase at different rates? Diving to the experimental design, we have two different treatments over 32 days, as you can see in the picture. And the x-axis, we have the transfer, which are the duration of the experiment, which is 32 days. And in the y-axis, we have the temperature, starting on approximately from 45 to 50. And our first treatment would be the gradual treatment, starting the heat shocks from 45 and increasing by the end of the experiment to 50 degrees Celsius. And the second one would be the sudden treatment. The heat shocks are constantly on the 50 degrees Celsius. So the heat shocks are given on each transfer, and after each transfer, the phages are grown under a specific host at 25 degrees Celsius. As a hypothesis, we expect in the sudden treatment population will acquire mutation at the earlier stages rather than the later, and in the gradual treatment, we expect the mutation will be acquired during, all over the experiment. The reason that we're expecting this result is in the sudden treatment, the environment is more stressful for the phages, and we expect them to accumulate the mutation at the earlier stages to be survived by the end of the experiment. But in the gradual treatment, the stress is lower in compared to the sudden treatment, and also the temperature is changing all over the experiment, and the phages are exposed to new environment. So we expect them to mutate, to have mutations all over the experiment. So now we are finished with the experimental design and moving to the methodology and sequencing. So we are looking at the mutations for the evolution experiment. First of all, we need to extract the RNA and purify the genome. And then we need to reverse transcript it through cDNA for the further steps, and then using PCR to amplifying the genome that we are aiming to study. And to make sure that we have the genetic material, we need to visualize them with gel electrophoresis and sequencing the genome. So since we are using the sequencing, we need to targeting and identifying the mutation. As you can see in the picture, on the outer membrane, on the outer surface, we have the membrane of the virus, which is originated from the host, as I mentioned before. In the middle part, we have the protein code, and in the inner part, we have the viral genome. 5-6 has a segmented viral genome, and it has 3 segments: small, medium, and large. So for detecting the mutations, we are focusing on 2 genes. Gene 8, which encodes the P8 protein, which is a shell protein and representing the capsid of the 5-6 structure, and gene 5 encodes the P5 protein, lysis protein. Lysis protein is essential for digesting the host membrane and inserting the viral genome inside the host. So these 2 genes are important because they are essential for viral infection. In terms of collecting the data, we faced some troubleshooting in terms of reverse transcription. We have one successful gel electrophoresis using 1-day-old prototranscript2 enzyme. And as you can see in the picture, here we have our samples, S1D16, S1D24, and S1D16 again. And we had the band, but after that, we had unsuccessful gel electrophoresis when we were using 1-week-old prototranscript2 enzyme. And as you can see, we didn't have the bands. However, we had the bands on our positive control. But still, we have some primary data from 2 lineages of the gradual and sudden treatment. As you can see in the table, We have the data related to, again, gene P5 and P8, and also considering both sudden and gradual treatment. Hopefully, this data would help us to identify the mutation, the timing the mutation arose, or about the differences of the mutation in different treatments. As an alternative way to sequencing the genome, now we are trying to use nanopore sequencing. Nanopore sequencing is a PCR-free sequencing technique. With this technique, we are able to sequence RNA directly, and since this technique and kits are dedicated to study just single-stranded RNA, now we are trying to develop our own protocol for double-stranded RNA, since the 5/6 is double-stranded RNA. So as our future work, we hopefully can sequence the entire genome at once with the help of the nanopore sequencing. And while we got the data, we want to know, does the rate of temperature increase matter in bacteriophage evolution or not, with the help of identifying different mutations and different numbers of the mutations in 2 different treatments. And also we want to know about the specific timing of the accumulation of the mutations. Thanks for your attention, and I would answer any question if I have time.
Thank you, Panyan. Are there any questions? Maybe if you can stop sharing your screen, we can see better. Okay, I then have a question. So when you do the evolution treatment with heat shock, do you always have adaptation? I mean, are you losing also some population that fail to adapt?
When we are, you mean on each transfer?
Yeah, do you lose sometime your?
Yes, we lose some of them, and when after heat shock, we grow them on the host, and after purifying of the genome, also we grow them on the host to know about the survival of the virus, and also knowing about the virus concentration after heat shock. Yeah.
So what's the fraction that you lose?
Sorry, what was that?
Sorry. So how many times do you actually the virus are really killed by or go extinct?
Typically 32 transfers, and on each transfer we grow them twice.
Okay.
Hopefully answered your question. Yeah.
Okay. So if we have no more questions, I would like to thank all the speakers for excellent talks this session, and I guess there's no break, and see you to the next sessions. Thank you all.